Assalamu alaikum and a very good day. I am Dr. Kinza Rafi, currently working as an assistant professor in Faculty of Engineering, Sciences and Technology, Hamdard University, main campus, Karachi. Here, I am going to present my research work entitled N2-hydroxyphenylacetamide protects against phytohemagglutinin-induced Alzheimer's disease-like pathology in SHSY55 cells that has been conducted in International Center for Chemical and Biological Sciences, University of Karachi. Starting with the introduction of Alzheimer's disease. Alzheimer's disease was first discovered in 1906 as an unusual disease of the cerebral cortex by a pathologist named Alois Alzheimer after examination of a female autopsy who died because of progressive cognitive and behavioral disorder. Clinically, Alzheimer's disease is defined by the decline of memory and cognition, while pathologically it is characterized by the presence of two abnormal structures in the brain that are extracellularly formed amyloid beta plaques and intracellularly present neurofibrillary tangles. Alzheimer's is mainly divided into 4 stages that are mild cognitive impairment, mild Alzheimer's, moderate Alzheimer's, and severe Alzheimer's. All these stages of the disease affect different areas of the brain. The disease symptoms begins with dementia and progress through the stages differently depending upon the brain area affected. There are both genetic and environmental factors that contribute to the development of Alzheimer's disease, but the major factor among all is age. Now, moving to the significance of the work. Currently, the treatment approaches available for Alzheimer's disease are limited to acetylcholine esterase inhibitors and NMDA receptor antagonists that only provide symptomatic relief to the suffering individuals. Clinical trials are also underway to develop new Alzheimer's disease drugs, and the therapy is based on the use of non-steroidal anti-inflammatory drugs, antioxidants, monoclonal antibodies, and kinase inhibitors. Considering the ineffectiveness of the available drugs, the study was designed to develop in vitro model of Alzheimer's disease-like pathology by using the plant-lectin phytohemagglutinin that triggers cells to release inflammatory molecules as neuroinflammation is initially involved in the onset and progression of Alzheimer's disease. Further, the study was designed to evaluate the effectiveness of the test compound Na2 against Alzheimer's disease pathology. Na2 is an acetamide analog and has been previously reported to possess anti-inflammatory and antioxidant studies. The study was initiated by the development of in vitro model of AD. To achieve this goal, human neuroblastoma SHSY55 cells were stimulated with different concentrations of PHA and were observed through phase contrast microscopy for the presence of neuronal aggregates. Further, reactive oxygen species formation was determined in PHA-induced cells by fluorimetric method using the H2DCFDA dye. After confirming the role of PHA in cellular aggregation and ROS production, Na2 was tested against the SHSY55 cells for measurement of cell viability using MTT assay. The neuroprotective effect of Na2 was also evaluated against PHA-induced cells through microscopy, and its role in decreasing reactive oxygen species was also determined by the same mentioned fluorimetric method. Moreover, the role of Na2 in reducing neuroinflammation and Alzheimer's disease associated disease markers was confirmed by PCR and immunostaining. Now, moving to the findings of the study. To observe the effect of Na2 on the viability of SHSY55 cells, 100, 200, and 300 micromolar concentrations of Na2 were used, and it was found that among all these concentrations, 200 micromolar concentration of Na2 showed maximum percentage viability, which is 141.18% plus minus 8.54 standard error of the mean. Based on the result of MTT assay, it was decided to use 200 micromolar of Na2 for further experiments. Next, to evaluate the effectiveness of Na2 against PHA-induced cellular damage, the cells were stimulated and treated in different manner. In the first experiment, the cells were treated with Na2 for 48 hours and then stimulated with PHA for 24 hours. In the second experiment, the cells were first stimulated with PHA for 24 hours, then treated with 200 micromolar concentration of Na2 for 48 hours. In the third experiment, the cells were first treated with Na2 for 24 hours, and then after that, without removing the compound Na2, the cells were stimulated with PHA for 24 hours. In all these experiments and the subsequent experiments, the concentration of PHA used was 10 micromolar, as it was the concentration that produced maximum loss in SHSY55 cells upon stimulation. From the results of phase contrast microscopy, it was found that Na2 was only effective and restored cellular deterioration. The cells were post-treated with Na2 as shown here. Then the effect of Na2 on reactive oxygen species formation was determined, and it was observed that Na2 reduced the production of ROS in PHA stimulated cells. Here, H2O2 was used as a positive indicator for ROS production, and donepezil was used as Alzheimer's disease standard drug. Next, the mRNA expression of inflammatory molecules was assessed in response to Na2 treatment, and it was found that Na2 downregulated the expression of interleukin 1 beta, tumor necrosis factor alpha, P38 alpha, and P38 beta genes that was increased as a result of PHA stimulation. Further, Na2 was found effective in reducing the expression of BACE1 gene, that is a beta secretase and is involved in the generation of pathogenic amyloid beta peptide. But it was not found effective in increasing the level of ADAM10 or alpha secretase gene that is involved in the non-amyloidogenic pathway. Moreover, the effect of Na2 was observed on the protein expression of amyloid beta peptide, mitogen activated protein kinase P38, which is involved in inflammatory pathways, and phosphorylated tau protein. From the results of immunocytochemistry, it was found that Na2 decreased the expression of amyloid beta peptide, evident from reduced immunofluorescence in Na2 treated cells. Further, Na2 reduced the protein expression of MAPK P38 as shown here, and the expression of phosphorylated tau protein, which demonstrated the role of Na2 in reducing the levels of Alzheimer's disease associated pathological markers. So, the study concluded that PHA is involved in amyloid beta overproduction and tau protein hyperphosphorylation in neuroblastoma SHSY55 cells. Also, Na2 is increasing the viability of cells and morphologically subsiding the effects of PHA-induced cellular damage. Na2 is inhibiting ROS and downregulating the expression of interleukin 1 beta, TNF alpha, P38 alpha, and P38 beta genes. BASE1 expression is also downregulated by Na2 treatment. Moreover, Na2 is reducing amyloid beta, activated P38, and phosphorylated tau in immunostaining experiment. And thank you for listening the talk.